This lecture will cover human blood composition, the only fluid tissue in our bodies. I will be providing you with important information that will hopefully allow you a better understanding of cells that make up human blood. First, I will talk with you about plasma, the liquid medium that supports our blood cells. Then I will cover red blood cells and their importance as a life-sustaining force. Afterward, I will go over white blood cells and the important immune responses that each of these cells performs. Then after that, I will cover platelets and the important roles that they play in supporting the well-being of the cardiovascular system. As well, I will be sharing with you a video that actually details and shows exactly how platelets form a blood clot. I hope you found this lecture beneficial in understanding the makeup of blood and its many benefits. Let's begin. In covering the basics, it is important to note that blood is a connective tissue. Why is it considered a connective tissue, you may ask? Blood is a specialized type of connective tissue in which living blood cells, called the formed elements, are suspended in a non-living fluid matrix called plasma. The collagen and elastic fibers, typical of other connective tissues, are absent from blood, but dissolved fibrous proteins become visible as fibrin strands during blood clotting. Blood is mostly plasma, which is a liquid containing dissolved substances such as blood cells, hormones, water, antigens, and electrolytes, to name a few. You may be asking yourself or wondering why it is that blood is a liquid. And if you consider the idea of what it would be like to have a thicker substance pumping through your arteries and veins, you'll quickly realize that the consistency of blood as a liquid is crucial for proper function. Thick blood would create a lot of problems and would be inefficient as a pathway for blood cells to properly function as they are intended. Other components that make up blood, as mentioned before, include red blood cells, known as erythrocytes, white blood cells, known as leukocytes, and platelets, also known as thrombocytes. Let's take a closer look at plasma as we continue our journey into understanding blood composition. Plasma, as previously mentioned, is the fluid matrix for which blood travels. It makes up 55% of blood volume. 90% of plasma is made up of water, which comes as no surprise given that most of our body is made up of water. Now, here you'll notice there are three main components to consider when talking about plasma. First, there is albumin, which makes up 60% of plasma proteins and is produced in the liver. It acts as a carrier to shuttle certain molecules through the circulation and is the major blood protein contributing to plasma osmotic pressure. Globulins, which make up 36% of plasma proteins, also act as carrier proteins. There are three globulins to consider. First are the alpha globulins, which inhibit certain blood protease and inhibitor activity. Next are beta globulins, and lastly, there are gamma globulins, which are also known as antibodies. The final main component for which we will consider here is fibrinogen. It makes up 4% of the plasma proteins and is produced in the liver. When needed, fibrinogen forms fibrin threads to aid the blood in clotting for blood vessel repairs when vessels are torn or rupture. The next component of blood composition that we will cover are red blood cells. These are also known as erythrocytes and have a biconcave shape. Their biconcave shape is the result of the cell lacking a nucleus, which it expels as it matures. Red blood cells contain hemoglobin, which carries oxygen from the lungs to the rest of the body. These cells also pick up carbon dioxide along their journey and return the carbon dioxide to the lungs to be expelled when we breathe. They are able to carry these molecules because of hemoglobin. Each hemoglobin is made up of 4 polypeptide chains, 2 alpha and 2 beta, which contain 4 heme groups per molecule. Red blood cells take about 15 days to develop, and they live approximately 120 days, and they are produced in red bone marrow. Now, next up, we will talk about white blood cells. White blood cells, also known as leukocytes, fall into two categories. The first category that we will talk about are the granulocytes. These contain neutrophils, eosinophils, and basophils. Neutrophils are the most abundant of the white blood cells. They have a multi-lobed nucleus, cytoplasmic granules, and they are our first line of defense against bacteria. These cells phagocytize bacteria and typically develop over about 14 days. They have an average lifespan of about 6 hours to a few days. Then we have eosinophils. Their nucleus is bilobed. They have red cytoplasmic granules, and they're best known for killing parasitic worms, as well as having a complex role in allergy and asthma. Their development duration is about 14 days, and they have a lifespan of about 5 days. Lastly, when looking at the granulocytes, we'll be talking about basophils. They have a bilobed nucleus with large purplish-black cytoplasmic granules, which release histamine, contain heparin, and have development duration between 1 to 7 days, with a lifespan of a few hours to a few days. Next up, we'll be looking at the second group of white blood cells, known as the agranulocytes. The first one we will consider is the lymphocyte, which has a spherical nucleus, a pale blue cytoplasm, and typically mounts an immune response by direct cell attack or via antibodies. It has a development duration period of days to weeks and a lifespan of hours to years. And lastly, when looking at agranulocytes, we have the monocyte, which has a U or kidney-shaped nucleus, gray-blue cytoplasm, and these are known for their role in phagocytosis and are also capable of developing into macrophages in the tissues. Their development duration is 2 to 3 days and have a lifespan of a few months. When thinking of platelets, we may often feel as though this is something that takes place internally and is something we cannot see for ourselves. Yet if you scraped your body, such as your hands or your knees, and have seen a yellowy substance before, you've seen plasma and the benefits that platelets can offer. Such actions are seen when we develop a scab. It is in this moment that we see platelets up close and in action. It's important to note that platelets have a discoid cytoplasmic fragment containing granules. They have no nucleus and are smaller than red blood cells. As well, they're known for repairing blood vessels and initiating blood clotting. Their development duration is 4 to 5 days, with a lifespan between 5 to 10 days. And it's also important to note that platelets are not cells in the strict sense. They are cytoplasmic fragments of an extraordinarily large cell called the megakaryocyte. Formation is regulated by a hormone called thrombopoietin. Next, we'll be looking at a video that shows exactly how blood clots form. I hope that you enjoy.
Normally, the flow of blood through blood vessels is unimpeded. Red cells, white cells, and platelets pass over the unbroken lining of endothelial cells. Its smoothness discourages their adhesion. But there exists, within this supporting framework of proteins and mucopolysaccharides, an emergency mechanism capable of reducing blood loss by promoting coagulation. This is the extracellular matrix, or ECM. The first reaction to an injury involving the epithelium comes from the damaged epithelial cells themselves. They release endothelin, a polypeptide which causes smooth muscle in the wall of the artery to contract, as do nerve impulses arriving in response to the injury. As a result, less blood is lost because the vessel is narrowed at the site of injury. The exposed ECM releases von Willebrand factor. This attracts platelets to the damaged area and promotes adhesion between these platelets. It forms bridges between platelets and the underlying extracellular matrix. Von Willebrand factor is also found on the surface of epithelial cells and in platelets as alpha granules. As platelets adhere to one another and to the extracellular matrix thanks to von Willebrand factor, they become activated, causing a reorganization of their actin fibers and a change in their shape. This in turn exposes their lipoprotein surface to the blood clotting factors. The activated platelets now release adenosine diphosphate, or ADP, and increase the synthesis of thromboxane A2, both of which boost platelet attraction. Both of these molecules attract more platelets to the injury site, expanding the platelet mass and further plugging the breach. Secondary hemostasis is the process of stabilizing the clot. It begins with the coagulation cascade, a sequence of events which develops with explosive speed, ending in the formation of a fibrin clot. It starts when factor VII binds to tissue factor. This activated complex attaches itself to the platelet surface, setting the cascade in motion. It activates factor X so that there is a rapid increase in the level of activated factor X, 10A, in the blood. Activated factor X cleaves prothrombin, yielding thrombin. In turn, thrombin cleaves fibrinogen to form strands of fibrin. We have been tracing the rapid response pathway of clot formation, from injury to the cells lining blood vessels through to the formation of the fibrin network. This pathway is known as the extrinsic system. But there is another slower pathway called the intrinsic system, which may be triggered by exposure of blood to a negatively charged surface, such as collagen or glass. The two systems are indeed complementary pathways which join at the level of activation of factor X. Polymerized fibrin forms a network which anchors blood cells and platelets in the clot.
That brings a close to this lecture on the composition of human blood. I hope that you've enjoyed this video. Feel free to leave a comment or suggestions, and good luck in your studies.